Hi, I'm Paul. I'm assistant professor in Department of Biomedical Engineering here in Changchun University. Today, I'd like to use this YouTube clip to introduce our lab's AI cross with microfluidics laboratory. A brief introduction to myself. I did my bachelor in clinical laboratory science, and then I started to study microfluidics and microscopy starting from master. I did my master in biophotonics in Yangming University, and then I did my PhD in Okinawa Institute of Science and Technology in 2020. Afterward, I did a couple years of postdoc focusing on commercialization of deep learning technologies and founded a small startup company. I started my current position as assistant professor from 2022. Primarily, my research interest lies in microenvironment, where this requires integration at the junction between basic biology as well as clinical needs and application as well as realization using engineering technique. So my research specialty lies with microfabrication, lab on chips, biophotonics, and clinical laboratory science. My research interest falls on building microfluidic platforms to study microenvironments, so specifically combination of multiple different materials, what I call heterogeneous microfluidics prototyping. And primarily, we want to develop organ-on-chip platforms for personalized medicine. Now, this also requires a development of technologies where we can quantify as well as analyze detailed information on cells or other model organisms on these chip platform, and we're gonna utilize deep learning to help us do that. So specifically on spatial temporal cellular dynamics analysis, and we're also interested in developing some of the quantitative microscopy techniques where we can realize that. So in addition, we're using microfluidic systems to study biomechanics in microenvironments. So a brief introduction to some of the key outcomes of our research over the past few years, one of which is development of heterogeneous microfluidic devices. We know there are several materials that commonly used for fabrication of microfluidics, plastics, silicone, rubber, or glass, and all of these, they each have their own pros and cons. And by combination of different materials, which we want to maximize each of their advantages and mitigate some of their cons. So for one is that we can quickly prototype depending on our study topics, depending on our needs. We can use engineering techniques to develop chips that can help us establish chemical gradients or physical stimulation much more easier in heterogeneous microfluidic devices. For example, we can build this that is better as a better alternative to conventional platforms, or we can customize, let's say, reversibly sealable microfluidic devices for various applications. Now, in addition, understanding the thermodynamics and fundamental fluid dynamics inside microfluidic systems help us a lot, and this is something we can also realize by heterogeneous microfluidic devices, where if it were a conventional typical microfluidic devices, if there's any hydrostatic pressure differences, we inevitably have secondary flow that could displace the cells and then cause them to aggregate. Now, this will be a disaster for analysis as well as cell experiments, where we cannot get single cell behaviors, nor could we analyze this reliably. Now, by understanding through first principle how this happens, we can easily design a microfluidic systems where we can mitigate out these hydrostatic pressure difference. Without secondary flow, all the cells, we only need very minute amount of cells to occupy the microfluidic channel, and the cells will be very uniform. So we can analyze single cell behaviors in microfluidic systems. One of our key research topic is on adapting deep learning techniques or these artificial neural networks for spatial temporal data analysis on our cell microscopy data. So this will enable more accurate analysis on displacement, speed, as well as morphology changes. Here on the left, you can see a typical phase contrast microscope images of cells, and these cells are undergoing stimulation on microfluidic chips, where they move toward the left and they actually have these morphological changes. These images are quite vivid for humans, but this is actually quite challenging for computer to analyze this kind of data. Typically, for conventional computer vision algorithm, they have trouble getting very accurate outlines. And moreover, is that for cells in high density, for example, here, most of the conventional computer vision software will recognize this as a huge blob and cannot give you more detailed analysis. By adapting deep learning techniques over the past few years, we know that we can use artificial neural network to recognize single cells. Even in these highly populated area, we can segment them accurately, and moreover, we can also classify them based on their morphology. For example, cells undergoing mitosis can be recognized here as well. And moreover, is that artificial neural network give us unique opportunities to predict based on how objects look like. So if we were training the neural network to recognize some of the more advanced phenotypes such as cell cycle or cell senescence, the artificial neural network, upon proper training, they can do prediction based on only fresh phase contrast images. So conventionally, these more complex phenotype requires you to fix the cells, that meaning you kill the cells and then stain them for some specific dyes, analyze them through other techniques. But eventually, in that essence, is that you get the snapshot of the live cells when you fix that. But these cells are already gone. You cannot reculture them. You cannot do any other process. But by enabling these phenotype prediction using artificial neural network, we can actually predict them while they're still alive, and then we can do appropriate treatments. Let's say if the morphology or the phenotype looks a bit different. That is the unique opportunities that could only be offered by deep learning or artificial neural network compared to conventional expert system algorithm. In addition, is that we focus a lot on how to analyze the data. Turning these complex images into something analyzable is, of course, one of the bottleneck. But eventually, is our customers or our user want is that we take images from microscope, but we ultimately want quantified data and statistically comparable data. And this is something we can actually combine to automate in a pipeline that is powered by artificial intelligence. Now, eventually is that this right now we can have multiple different advanced automated system where we can collect a lot of data from gigabytes to terabytes per day very, very easily. Now, the problem is the bottleneck lies in how to analyze the data. Let's say today, if I want to track unstained cells fully in both their displacement as well as their morphology changes, the fact is that oftentimes a student, assistant, or even the professor needs to do manual analysis on each of the cells, where you need to outline the cells and then for each cells in each time frame throughout the time. So oftentimes this one field of view data oftentimes can amount to 4 to 5 hours of work, even if you're well experienced and you're using a tablet. There are some software on the market that is based on conventional computer vision. Somewhat can automate it, but oftentimes the problem for this is that they will generate lots of we call false tracklets, basically false data that could also mask some of your real data if there's even any. So that means you need to spend a lot of time also manually to look over the data and exclude those mistakes that computer make. There are also some newer startups that's offering software as a service, where you upload the software that is being processed by a proprietary closed source algorithm on the cloud, and then you download the final data. Now, the problem is the uniqueness for this space is the data from microscope. The images are often very, very large. You will spend very significant time uploading the data from local machines to the cloud. In addition, the instances on the cloud may have various performance where you can range from processing in 5 minutes or ranges processing with hours. In addition, they still have that false tracklet problems where you need to spend some time manually to process, to verify or even edit the final data. What our essence for Udine is, we create this artificial intelligence or deep learning powered pipeline where we can automate this process, turn 4 to 5 hours of work within 5 minutes, all the way from segmentation to tracking to analysis. And in future, we want to even reel out some of these labor, some of the problems caused by additional data transfer that will require labor effort. So we can achieve 6 to 60 times faster results. In addition, accuracy is also a key. The problem for manual not only is daunting task, you also inevitably introduce some of the human errors where the oftentimes experimenter and the data analyst is the same guy. So this inevitably causes some problems where observer bias could be introduced, which could cause some problems. Now, the problem for the software now on the market is, like I said, with a lot of false tracklets that will easily mask the real data that you could get. Oftentimes, not only just masking, oftentimes these false tracklets introduce completely opposite results, and thereby bringing your accuracy down from 30% to 60%. Now, our system with high accuracy of instant segmentation offered by the artificial neural network, we can get very, very high accuracy, and with simple software algorithm limits, we can also bring it up to very close to a human would do. And in addition, this we can easily process a lot of data and completely bias free from human errors. So we can get 150% up to 400% more accurate compared to existing solutions. Now, based on this, what our goal is to develop, innovate new laboratory systems by integrating AI software with edge computing system. Not only we're developing software as a cloud solution like others, we also want to develop a smart microscope where AI software is integrated with edge computing, where we can take images and automatically do instant data analysis powered by AI. Now, we interviewed a lot of people, and the common thing shared by biomedical or engineering experimenter is that we oftentimes work on different things, but we share a need that we need to take images to document our findings. But oftentimes is that taking images document findings, you still need a person to do post hoc analysis afterward, which is oftentimes daunting task and very time consuming task. What we want to do is put our imaging systems integrate that with AI software to offer us instant data analysis, where immediate data results can that experimenter decide what things to do when they're doing the experiments. Maybe they want to do immediate control, maybe they want to do immediate addition of different reagents. In addition, by further incorporating Internet of Things, the internet connectivity to our microscope, we can also enable that you don't have to be inside the lab to check how your sample looks like. For example, early time of COVID times, many of the university in Japan and Europe, they are restricting the online personnel inside the lab. Oftentimes, we also wonder, we cannot do anything until the sample is ready. But we cannot know that right now until we go into the lab and actually check up upon the samples. But with this vision is that this kind of machine can enable you inside your home or your office. You can check how the sample looks like in the lab and decide whether or not you do need to go in and commute and go into the lab to start the experiments. So we also have other smart integrated platforms that we think that we can integrate them into automated system. Now, in addition, for microfluidic, come back to the microfluidic part, we can design complex multi-level microfluidic chips as platforms for any other basic biology. For example, these you can see are microfluidic channels ranging from 800 nanometers to a couple microns thick, and then the width ranges from 1 micron to 3 microns. In these microchannels, we can trap individual bacteria and then observe how they outgrow and then compete as well as evolve with each other. Okay, so here you see these are bacteria, actually the E. coli, similar to what we can find in human gut, and we transfect them with two different fluorescents, one green, one red, and inside microchannels. Through our very long time lapse, we can see how they compete. They align inside confined geometry. So this is a powerfulness of microfluidic system, especially we can tailor design for different experiments. Our current research direction primarily lies on developing novel organ-on-chip system, specifically for cardiovascular and brain tumor biology. Moreover, we focus on building blood vessel platforms where we can use it as a platform to mimic humans in drug discovery. And this lies very core to some of the mainstream international standards and direction right now. For example, US FDA as well as EU's Horizon Europe, as well as international ICH organization, many of whom are pushing for the 3Rs principles, where we want to reduce the use of animal models, and eventually is the data, the drug discovery data you found on animal models, they could still have gaps with humans clinically. So by using in vitro, by using organ-on-chip, this can be a very potentially very useful replacement for animal models in future. And this is something that we invest a lot on here. The other one is that we can also use microfluidic technology for basic applications, basic biology studies, and application in tissue engineering, where we can use microfluidic systems and create a complex design that we can incorporate physical as well as chemical stimulation, so that we can see how cells as well as tissues evolve or mature upon these platforms. And then maybe these will carry a lot of application in potential clinical needs as well as tissue engineering. Now the third part is on pushing for our analysis of all the data on these microfluidic platforms. So we want to create an AI microscope as well as quantitative microscopy techniques to help us quantify and also better track how cells and tissue mature on microfluidic systems. So we have many more projects. Our laboratory resources lies on BSL-1 facilities, incubator, as well as BSC biosafety cabinet. We also build a lot of prototypes, basically epifluorescence microscopes. We also design ourselves and modify it so that it can incorporate our customized microfluidic systems and enable fully modularization. We also have electronics and machining workstation to help us help our various prototyping efforts. CO2 laser cutter for fabrication of plastic microfluidic devices. A deep learning CPU workstation also sponsored a lot, supported by NVIDIA, and 3D modeling various 3D modeling software to help us model the system. In our department, the core facility includes various of FDM 3D printers, as well as a nylon powder laser sintering 3D printer, where we can actually turn a lot of complex design. Some of those is impossible to be made by conventional CNC machining, or very expensive if they were made by CNC. We can easily realize and prototype those using laser sintering nylons, and the parts will be very strong and biocompatible and relatively much lower cost compared to machining. In addition, we fabricate a lot of microfluidic devices where many of the microfabrication process come in. We actually have a massless writer and dual wavelength so that we can fabricate the devices directly or fabricate the photomask that are required to fabricate these micro devices. So what can you learn inside our lab? For one is, of course, fabrication of microfluidics devices based on different materials, for example, plastics or silicone rubber, as well as the microfabrication that required for this. Our lab is also very interdisciplinary. We will have application in cells or bacteria or various model organisms, so you can also learn biomedical techniques as well as culture for these bacteria or cells. We also work on microscope a lot. In addition, we prototype a lot of things, so 3D modeling and printing as well as basic electronics machining techniques is something that you can learn in our lab. In addition, we focus a lot on deep learning applications, more core to biomedical, applied to biomedical image analysis. So this is also one of the fields that you can also learn inside the lab. In addition, we follow strongly on lean startup techniques. For example, turning based on problems, we find solutions, and based on those, we develop solutions for targeting these problems. So for interested students, you're welcome to join. If you want more info, please visit our lab website. You can contact me through email or find contact page on website. So our lab core value is respect, honesty, and especially grit. So are you a hands-on person, or if you just want to learn the techniques such as microfluidics, microfabrication, cell culture, or if you have your own idea that you just want to try, you're welcome to contact us and see potential joining us, especially if you are interested in collaborating on data science, AI, or electromechanics integration. Now, if you have goals or plans to study abroad or build your own startup, you're also welcome to reach out so we can interact with each other and learn from each other as well. So, thank you.